Okay, thank you very much. The next section is going to be theme 2, and it'll be led by Yana. And she has also 3 presenters, as all 3 themes have. And so, Yana, take it away.
Hi, thanks. Good to see everybody on this second day. Also awesome talks so far. I'm going to talk, well, I'm not going to talk, but my postdocs are going to talk about all the things that they've been doing. The first person to talk about the things happening in theme 2 is Ariel Aptekmann. Ariel got his PhD in biological chemistry from the University of Buenos Aires in Argentina. And he joined the lab at the very beginning of this ENIGMA project, pretty much 2 years ago, working joint between the Ferreira lab in UVA and my lab at Rutgers. And his primary efforts are to predict metal binding, preferably from reads that you could sequence from metagenomic data. And also, he is looking to identify the repeat tiles and protein structures, which I talked about yesterday. And to do that, he would be able to then tell us which motifs, in structural motifs, would have been around at the origins of life. So Ariel, do you want to set up and start?
Today I'm going to talk about ancient protein repeats. Although this is part of theme 2, it is heavily overlap with things that people have been talking in theme 1, like what Vic and Saroj have been talking about. First of all, I want to show you that there are repeats that you can find in proteins. That means sort of symmetries that can be easily observed. And those are essentially pieces of structure that repeat themselves within a protein. These repeats occur only in structure. And if I take the pieces of each one of these repeats and I extract the sequence, which is what I'm doing down here, you can see that there is very little sequence conservation. So these repeats are not easily identified in sequence. People have observed all the repeats that you can find in a family, and they found that a lot of repeats occur with very low sequence identity, which is what I'm showing here. And most of them are occurring among around 30% sequence identity, which is below what you can find with normal sequence alignment methods. So how do we find what is the repetitive unit? For this, I'm going to take as an example this painting from Escher called Birdfish. And we could break this by using several different axes of symmetry. For example, we could use this piece, the blue tile, which has 2 repeats, but we could also break this same painting with the red tile, which also has 2 possible tilings. And we could actually define many more that would have several occurrences within this painting. So how do we scan for these repetitions? We can take essentially 2 parameters, a size for a window in sequence space and a location for that window in sequence space, which would be analogous to the size of the square and the position of that square in the painting that we were talking about. We developed a tool that uses some criteria to define what is a tile, one of those repetitive units. The first important tiling criteria that we use is that we will only accept those tiles that do not overlap. And then we have a scoring function, which reflects the coverage of the tile on the sequence space, taking out the trivial coverage that it has, because any piece of the protein always is at least one time within that protein. So what we want to know is how much of what is not the original tile we can cover with that tile. And that is the tile score. So when we proceed to break the protein structure into pieces and find where in the structure those pieces are repeated. If we have, for example, only one repetition, we will see that enlarging the size of that window won't affect the score, which is what we see on this n=1 line. If we have two repetitions, the larger the window size is, the more of the sequence is going to be covered. And that is this line over here. This is if we have 3 repetitions, this is what will occur if we have 4, and so forth. On the other hand, if we consider the tile score, the relation with the size of that window, the larger the window is, the more of the repetition that we're going to cover, until a point where we are no longer covering that repetition, but we're starting to go outside of that repetition, and therefore the score will drop. So it is very useful to have the tile delta, which is what is plotted down here on the dotted line that has a small peak, because that's indicating that that is the proper window size for finding a repetition. Maybe this is better shown with an example. Here we are visualizing the tessellation for a protein. What we have here is the tile length, that is window size we are using to break this protein, and what is the position of each one of these pieces. And the color represents the tile score, which is what I explained before, the coverage of that fragment for the protein. For things that are around size 10, we have fragments that are these alpha helixes, of which I have 8 occurrences on the protein. And that essentially is showing us that at that size, we are only picking up something that is a piece of alpha helixes, 2 terms. If we go to smaller sizes, we are only going to pick up those, alpha helixes and beta. If we go up, we are going to find each one of these 4 repetitions, but without the loop. And if we go to a tile size of 33, approximately, we have a repeat that we can displace the position of the start, and we are always tiling a large amount of the protein. That thing we can identify as a wave. We are going to call that as a wave, because we can see that as a score that remains high no matter the position of the tile center. So I wanted to show you several classical repeat proteins, and here on the side is plotted the delta of the tile score, and we can see clear peaks at the sizes where we have a repetitive unit. If we have a repetitive unit of size, let's say 33, which is the typical for ankyrin, we're going to also have a peak at size 66, which is picking up 2 of the smaller scale repetitions. And we're also going to have the same at other harmonic lengths. But not all proteins are repetitive. There are some that don't hold any repetitive signal, like the one on the bottom right. Or actually, most of these don't have very clear repeats. There is some peak in some of them, but we cannot say that they are as repetitive as the other ones that I showed before. So what we are going to try to do is extract each one of these tiles by identifying the peaks on the tile delta score for each window size. And this is telling me that at size, let's say 30, there is a clear peak. So what we're going to do is we're going to extract the maximal score for that window size. To keep that as a tile that comes from this protein. And this whole process that we developed works as a tool, and I think it's very interesting because it might actually be possible to combine this with the work that other people on ENIGMA is doing right now, and use it to identify repetitive pieces. But we can talk about that later if someone is interested. My idea is that this is going to be available tool at some point. All right, so this was the repeat part of the story, and now I'm going to tell you about the ancient part of the story. So as Jana mentioned yesterday, she developed a network, a similarity network of all the metal spheres. She then clustered those and obtained a smaller network of around 1,500 nodes. And from those, she selected 15 nodes, which are the most central ones, and that are also possibly the most likely ones to be at the origin of metal binding. So what I did was identify the repeats for each one of these. This is one example. I'm going to explain this with one example, and at the end show you the results for all of them. I took each one of those proteins that Jana gave us as the possibly most ancient metal binders and tried to identify tiles within them. And for each one of these tiles, and for the example that I'm showing you, we're going to follow this one, the size 20. We took each one of those tiles. Here, that tile is colored in red on this structure. And we did a cross-tiling. That means taking that tile and seeing how well does it cover the structure of another protein. And I'm showing you here an example. There are two occurrences overlapping very close to the metal binding site on a very different protein. That's the one in red and the one in green. To me, this is very interesting. I know that in this picture they look the same color, but the one on the left is a cobalt binding protein, and the one on the right is an iron binding protein. And although this sort of motif or fold is a little bit off and is not directly binding the iron ion, it is really close. And by looking into this kind of information, one can start to imagine evolutionary processes. So we did this for each one of the tiles that come out of each structure, and we tiled them across all the different structures. And what I'm showing you here is the tile coverage for each one of those. So this thing that is pointed with an arrow is the example that I just showed you. This is that fragment that was around the cobalt metal center and is also around the iron metal center on 3DHZ. So what do we do with this? Well, one approach could be to look at the highest scoring ones among these and take a closer look and use the best scoring ones as possible candidates for something very small that actually has metal binding capabilities. And another possibility is to see how well each one of these tiles covers the metal spheres, that is in the context of the spheres that Jana pointed out before, how well each one of these covers. And this is what I'm showing you. And there is a huge difference because some of these tiles cover a lot of the metal centers and some don't. And although this is not a finished work, my idea is that we can take this information and look it in detail for the best scoring ones and use that as a design tool so that a peak can actually design minimal binding faults. Anyway, the conclusions are that Tillingator, that's how I call that tool that I showed you at the beginning, can help you find repetitive patterns within proteins and across proteins also, maybe. And that the tiles that cover most of the structures are the best candidates to be ancient because they are conserved among the most ancient structures. And the tiles that cover the most spheres are candidates as the origin of metal binding. I would like to thank Jana and Diego, who helped me with this work and on each one of the parts that I mentioned. Also Rutgers and NASA for funding this research. the people at Bernberg Lab who always helps to discuss, and Paul, Vikas, and Akif, because they are part of this work, and they really helped a lot. So thank you very much. I don't know how much time did it take. Was that okay?
You are okay. We have a minute or two for questions if anyone has any. Doesn't look like.
This is Yilu here. It's very interesting and exciting work. I'm wondering from the perspective of the secondary spheres. I'm wondering this kind of tile approach, how easy, how ready can you come out with not only the primary sphere but also the second sphere? Does it cover that?
Yes. What I'm sure you're talking about this. This is the coverage on the secondary sphere.
Okay. And how much in terms of conserved? I guess the one with the highest, the most intense one are the most conserved than others. In other words, how do we inform this one for protein design engineering, like Akif?
Well, you could think of this in terms of structure conservation. The horizontals, the more conserved ones are going to look like very clear horizontal lines. That's pretty much the approach we could take on this one. Another approach would be to compare tiles and build sort of a tile tree and cluster them, and then have the ones that have the highest score.
What about to the function, like redox potential activities? How far can we go push this envelope?
That would be great, but I think for that we would need to take all of them and actually do some bench experiments, like physical experiments.
But can we actually get the redox potential or activity information map onto this tile coverage?
From the original one, sure. Yes, we could do that.
All right, thanks.
So this is exactly what one of our undergraduate students who's working with Kenneth is doing, Nolan. He's taking an old data set that was done by another undergraduate who went on to become an astrophysicist. But he has done an Excel spreadsheet and of all the redox potentials of the oxidoreductases that we know of to a common pH. And we're trying to map those to the obvious issue of the origin of proteins. For us, that's an obvious issue, but it's exactly right. We don't have that information very broadly available.
Yeah, we have some Excel sheet, but very limited one too. We just begin to accumulate that. So maybe you can share some of that together. Yeah. Thank you.
Yeah, actually, we'd be very happy to share that with you. And if you have students in your lab that can help us annotate that, or to add more proteins, because I don't think we have all your copper proteins in there. It would be fantastic. I think we would have a really very, very simple paper, which would be very useful to the community.
Yep, I agree. Yep, thanks.
I think we could also take whatever data you guys have and map it onto the particular proteins that you're interested in. I think Ariel's Tilingator tool runs fairly fast. And
yeah, so definitely we really like that, because I think that can guide us towards what the next mutation to do. Because although it's only one slide I mentioned about how to tune the potential, there's a lot of trial and errors, and many of them didn't work. Obviously, we don't publish it. The mutation, it didn't work. So I think having some guidance from this kind of computational insight is going to be extremely useful.
Well, we'll be looking forward to that. We have
Absolutely.
tools to analyze mutation sites and to predict which ones should be mutated if you're interested. So yes, let's talk about this.
Thank you.
Great. Thank you. Thank you, Ariel.